Nanoplasmon-Enhanced scattering, or NPAS, is a simple and non-invasive alternative to the surgical biopsy that can skip time-consuming and labor-intensive exosome purification steps, expanding the application of exosome analysis to clinical settings.
This NPAS assay is a rapid procedure to analyze specific biomarkers on the outer membrane of exosomes that does not require separate isolation and purification steps.
We only need a very small clinical sample for this assay. Therefore, this method can expand the use of NPAS to studying genetically engineered or PDX mouse models.
There are a few steps in this protocol that might seem daunting. However, with a few practice runs, everyone with basic wet lab skills can learn to successfully perform NPAS.
To begin the procedure, combine 40 microliters of a suspension of Nutravidin functionalized gold nanorods with 200 microliters of cold pH 7 phosphate buffered saline. Centrifuge the mixture at 8500 times G at 4 degrees Celsius for 10 minutes and remove the supernatant. Repeat this washing process twice more, and then resuspend the nanorods in 40 microliters of cold PBS. Next, add 150 microliters of cold PBS and 10 microliters of a 0.5 mg per milliliter solution of the appropriate biotinylated antibodies to the suspension. Mix at 4 degrees Celsius for 2 hours to obtain antibody-conjugated gold nanorods. Wash the nanorods 3 times by centrifugation in 200 microliter portions of cold PBS at 60